What is a phenotype? A phenotype is the set of observable characteristics of an organism resulting from the interaction between its genetic information and the environment. The relationship between genetics and phenotype is like a blueprint becoming a building. Your DNA contains instructions, but how those instructions are expressed depends on many factors. Phenotypes include a wide range of observable characteristics, such as physical traits like height and eye color, biochemical processes like enzyme activity, behaviors such as aggression or mating preferences, and even susceptibility to diseases. It's important to remember that phenotypes are what we can directly observe or measure, while genotypes are the underlying genetic instructions that may not be immediately visible. Genotype versus phenotype. What do these terms mean and how are they related? Genotype refers to the genetic makeup or DNA sequence of an organism. It's the genetic instructions that determine traits. Phenotype, on the other hand, refers to the observable characteristics or traits that result from the expression of those genes. Let's understand this with a simple example of eye color. The genes that code for eye color represent the genotype. Different gene variants determine whether a person will have brown or blue eyes. The actual eye color we observe, whether brown or blue, is the phenotype, the physical expression of those genes. Interestingly, different genotypes can sometimes produce the same phenotype. For example, in flowers, different genetic combinations can produce the same red color through entirely different biochemical pathways. Conversely, the same genotype can produce different phenotypes under different environmental conditions. For instance, Himalayan rabbits have a temperature-sensitive gene. In warm environments, they develop white fur all over their bodies. But in cooler environments, the same genetic code produces dark fur on their extremities, like ears, nose, and feet, all with the identical genetic code. In summary, genotype and phenotype have a complex relationship. While the genotype provides the genetic blueprint, the phenotype, what we actually observe, is influenced by both the genetic code and environmental factors. Environmental factors play a crucial role in how genes express themselves. The same genotype can produce different phenotypes depending on environmental conditions. Environmental factors affecting phenotypes can be categorized as internal and external. Internal factors include hormones, metabolism, epigenetic changes, and cell signaling pathways. External factors include temperature, light, diet and nutrition, and stress. Let's explore specific examples of how environment affects phenotypes. Nutrition significantly affects height. Two individuals with identical genetic potential for height can develop different adult heights based on their nutritional intake during development. In many reptile species, incubation temperature determines the sex of offspring. The same fertilized eggs develop into males at cooler temperatures and females at warmer temperatures, despite identical genetic makeup. Identical twins provide a perfect example of how environment affects phenotype. Despite having identical DNA, twins can develop different traits due to environmental differences. These differences can include height, weight, disease susceptibility, and even behavior patterns, all influenced by factors like diet, exercise, and other environmental exposures. Labrador retrievers provide a classic example of how multiple genes can influence a single trait. Labradors come in three main coat colors: black, chocolate, and yellow. These colors are determined by two different genes. The first gene is the B gene, which determines whether the pigment produced is black or brown. The dominant B allele produces black pigment, while the recessive B allele produces brown. The second gene is the E gene, which controls whether pigment is expressed at all. The dominant E allele allows pigment expression, while the recessive E allele prevents pigment expression, resulting in a yellow coat. When we combine these genes, we can understand how different genotypes produce the three coat colors. Dogs with at least one dominant B allele and at least one dominant E allele will have black coats. Dogs with two recessive B alleles, but at least one dominant E allele, will have chocolate coats. And any dog with two recessive E alleles will have a yellow coat, regardless of their B gene status. Let's look at how these two genes interact to produce a single phenotypic trait. The B gene determines the pigment color, while the E gene controls whether that pigment is expressed. Together, they determine the final coat color. In summary, the interplay between the B gene and the E gene demonstrates how multiple genes can work together to influence a single phenotypic trait. This example illustrates an important principle in genetics. Phenotypes are often the result of interactions between multiple genes, not just a single gene acting alone. Genes don't operate in isolation. Their effects depend on the environment they're exposed to. This is called gene-environment interaction. Gene-environment interactions occur when genetic variants respond differently to environmental factors. The same genotype can produce different phenotypes depending on environmental exposure. The interaction between genes and environment is complex. When genes and environmental factors interact, they produce unique effects that can't be predicted by looking at either one alone. Let's look at our first example, smoking and lung cancer risk. Some genetic variants significantly increase cancer risk in smokers, but show little effect in non-smokers. Our second example involves diet and weight-related genes. The FTO gene variant has been linked to obesity, but its effects can be neutralized by dietary choices. These examples help explain why genetic risk factors don't always lead to predicted outcomes. Genes don't act in isolation. Their effects depend on environmental context. The same genetic variant can lead to different outcomes in different environments. Medical applications of phenotype analysis. Doctors rely on observable symptoms, which are phenotypes, to diagnose medical conditions. These physical manifestations provide crucial diagnostic clues. Genetic testing allows us to connect specific genotypes to disease phenotypes. For example, testing can identify mutations in genes like BRCA1 that increase breast cancer risk. Personalized medicine tailors treatment to individuals based on their unique phenotypic variations. This approach recognizes that patients with the same disease may respond differently to treatments due to their genetic makeup. Pharmacogenomics studies how genetic differences affect drug responses. For example, variations in the CYP2D6 enzyme gene create different metabolizer phenotypes affecting how patients process certain medications. This genetic information allows clinicians to optimize drug selection and dosing, avoiding adverse effects and treatment failures. This application of phenotype analysis is transforming clinical practice. Phenotypic analysis drives crucial applications in agriculture and conservation biology. In agricultural breeding programs, farmers carefully select for desirable phenotypes to improve crop performance. These include high yield, with larger fruits and more seeds, disease resistance through enhanced immune responses, adaptation to different climates, like drought tolerance, and quality traits such as flavor, nutrition, and shelf life. Conservation biologists monitor phenotypic diversity in wild populations as an indicator of genetic health and adaptive potential. They track population health through body condition and size variation, assess adaptive potential by measuring trait diversity, and monitor for inbreeding effects, which typically show as reduced phenotypic variation. Modern phenotyping techniques now allow for much more precise connections between observed traits and their genetic basis. These include phenomics for high-throughput trait measurement, remote sensing using drones to monitor crop health, genomic selection that uses DNA markers to predict traits, and CRISPR technology for precise trait modification. A powerful example of phenotype engineering is golden rice, which demonstrates how understanding the connection between genes and phenotypes can address global challenges. Scientists identified vitamin A deficiency as a major health problem in developing regions. By engineering rice to produce beta-carotene, they created a distinctive golden yellow phenotype in the rice grains. This biofortified crop has the potential to prevent blindness and death in vulnerable populations.